I was excited to see our work on the cover of Neuromodulation, Technology, and the Neural Interface in this August 2025 issue. This work is actually a computational model of spinal cord stimulation. And it's considering how, when you activate spinal cord stimulation, current is delivered into the spinal cord. But we were especially interested in the current that gets into the dorsal horn. So not just thinking about the axons, the dorsal column axons that are the typical target, but saying, well, but the current's going to go through these dorsal columns and it's going to actually enter the dorsal horn. And while the amount that enters the dorsal horn is less than the amount that enters the dorsal column, it's still a significant amount. And in the dorsal horn, you have a variety of types of interneurons that we considered, including these very sort of wide islet type cells, vertical type cells, radial and central. And so the main question we had is, of the current during spinal cord stimulation that enters the spinal cord, so there is some current flow generated here, how much would these cells polarize, right? Will they polarize almost no millivolts, some millivolts, and so on? And so that's what we did. And I can briefly summarize what we found. Let's see here. Page 952. So it's a modeling paper, and there are certainly some technical aspects for those who are interested in those kind of details. One of the key technical innovations of this paper is our development of something that we're calling the mirror quasi-uniform estimate. What that basically means is that we made a lot of assumptions in order to simplify the process of the computational model, but understanding that is not relevant if you want to just interest in the big picture. Here's that same figure again in grayscale, where we are simulating, this is a CAD-derived model of the spinal cord, predicting how much current gets into the spinal cord, and then the resulting polarization. And so these are the kind of results that we're seeing here. So this is sort of the main figure here. And the main thing to understand is that we're considering these cell types independently, right? So here's the islet type, here's the vertical type. This is what these cells look like. And we're asking how much is each one of these polarized? And we're doing a bunch of different analyses here, thinking of both, under the electrode, between the electrode, thinking about directionality. But right here is the bottom line, where you're looking at each cell type, and we're asking what's the most that they might polarize. And what we found was that these islet cells, basically by virtue of how large they are, polarize the most. So this is 3.5 millivolts maximum polarization per milliamp applied. So 1 milliamp would produce 3.5 millivolt of polarization, while 2 milliamp could produce 7 millivolts of polarization. And this polarization was actually tended to be near these sort of dendritic tips. So the maximum polarization is not in the somas, but actually in the dendritic ends, while other cell types like the vertical type cells, we're not polarizing quite as much as the islet cells. And so this gives you a sense of how much polarization you get. But there is a caveat here. When we first did this analysis, we considered very long pulses, what we're calling sort of DC-like pulses. So these are pulses that will go on and will stay on for a very long time. But in reality, that's not how spinal cord stimulation is applied. Spinal cord stimulation is applied with very brief pulses. And this is the polarization for very long pulses. So we have to take this a step further, and that's what's shown in this figure here. And here we're looking at the pulse duration. And we're asking how much polarization do you get as a function of pulse duration? And you can see here that as the pulse durations decrease from 10 milliseconds, 1 milliseconds, and now getting into more typical SCS domains of 100 microseconds, the peak polarization that you see decreases. So whereas you might have seen for these islet cells up to 3.5 millivolts for very long pulses, as the pulses get shorter and shorter, the polarization you see gets less and less. And so that also means that longer pulses, all right, if you decide to use longer pulses, you will produce more polarization of these interneurons. And that is the main takeaway. Some current does get into the dorsal horn. That current will inevitably polarize the interneurons that are there. How much they polarize will certainly depend on the interneuron location as well as the type, and will strongly depend on the pulse duration that is used. And that's the paper. We also did some additional analysis thinking about the role of montage. So the analysis I showed you before were for two adjacent electrodes that had one gap between them, but you can move the electrodes from having one electrode gap between them to being perfectly adjacent. You can think about a tripolar montage, or you could think of a distant bipolar montage. And the question we're basically asking here is, what is the impact of the montage that you use on how much potential interneuron polarization you get? And so what we found is that here's, again, starting with that space bipolar where there's one gap in between the electrodes, and there's that 3.5 millivolt value. As you move the energized electrodes closer together, you will produce less polarization and also less for a tripolar montage. And so there is a moderate penalty for moving the electrodes closer together. If you move them farther apart, actually, there isn't much of a benefit. And so what that's telling us is by the time you have this sort of spaced bipolar montage, that's where you have an active, a gap, and another active, you're sort of producing the maximum polarization you're going to get of these dorsal horn interneurons without much benefit as far as peak polarization by moving the electrodes farther apart or even using a monopolar montage. However, there is a cost, so as you move the electrodes farther and farther apart, you will become less focal. While moving them closer together, you will become more focal, again, at a moderate cost of maximum polarization. And that's our paper.